Hello, this set of slides is just going to go over the first I will do in vitro fertilization using sea urchins as our model. So what you'll see in this stack of cards is some micrographs of some of the prepared slides that'll always be available in the lab. You know, they're stained, put in permanent slides, they're in racks. You can look at them anytime. Some of these micrographs are going to show developmental stages, but they're of live specimens, which of course won't keep. So to see those, you'll have to look at those, during lab while we're actually watching the embryos develop. All right, this is just a micrograph of an adult sea urchin. We're going to be using these as our models, so this is what we'll get the eggs and the sperm from. Now what you're seeing is the underside, the oral view of this sea urchin. So this side would normally be facing the substrate. These are in the phylum Echinodermata. They have a mesodermal skeleton. It is covered with softer tissue on the outside. So this whole thing, this big ball-like structure underneath would be a hard shell or test, it's called. Hard to get needles through. We'll use these to extract the eggs and the sperm. Where we'll do it, this area here, right next to the teeth, these are 5 interlocking teeth. Echinoderms typically have pentaradial symmetry based on sets of 5. But right next to the mouth here, this is a soft area where we can get in a needle. We'll inject some potassium chloride, which doesn't kill echinoderms, but it will cause them to release their eggs and their sperm, or eggs or sperm, depending on their sex. It's hard to tell their sex till they actually release their gametes. They all look the same. But we'll inject them here. The opening to the reproductive system is on the other end, or aboral end. It's also where the anus is. And the eggs and sperm will spew out. We'll collect them, put them together, watch fertilization in some of the early stages of development. Now this particular slide, this is slide number 3 of the presentation. This is showing you a prepared slide, so it's been stained, but it is dead, but the stain gives a little better contrast. And this is a slide of just an egg. This is an unfertilized egg as it would be released by a sea urchin. They're fairly small. You can see them with your naked eye, but they're fairly tiny eggs. And there's really not much to see. In here, this is just the kind of homogeneously mixed small amount of yolk in these eggs. So you can't really see any structure, just kind of this granular looking mixed yolk in here in the cytoplasm. Along the outside, there is, of course, a plasma membrane right around here somewhere. And outside of that, there is a vitelline envelope, which is a kind of a protein containing sort of capsule around the egg. Now, before fertilization, the vitelline envelope and the egg plasma membrane, they're fused together. There are proteins that hold them together, so you really can't even resolve them. I have a hard time, seeing which is which at this particular magnification and resolution. Now this is a slide. This is one of mine. I had to borrow it because I didn't have a good slide of a fresh unfertilized egg. But this is showing an unfertilized egg. It hasn't been processed or stained. So this one was a photograph taken while the egg was still viable. And again, you see the homogeneously mixed yolk in here. Along the outside would be the plasma membrane and the vitelline envelope, which again are fused together, so you can't really tell them apart. Now this slide number 5, this is a prepared specimen of an egg after it has been fertilized by a sperm. Now this one is, because it's been prepared, it's a little deformed. You get some artifacts introduced when you process these things. But again, you'll see the egg with its homogeneously mixed, yolk in here. That hasn't changed much. But what you want to notice is out here, you see this little sort of bag-like capsule or membrane around the egg. That used to be the vitelline envelope. But what happens after one sperm penetrates this egg and it releases its genetic material, that zygote, as it's called, that fertilized egg, has all the chromosomes it needs. But keep in mind, there can still be thousands of sperm out here. If another sperm gets in there and releases another set of chromosomes, it's referred to as polyspermy, that embryo won't be genetically viable. It'll have too many chromosomes and it won't develop normally. It will die. So in order to prevent that, these eggs have a protective mechanism. It's called slow block to polyspermy. We should go over it in lecture a bit. But basically, when that first sperm fertilizes the egg, there are some little cytoplasmic granules just on the inside of the egg's plasma membrane. Again, they're just little vesicles, little membrane-bound packets of material, mix of chemicals. When that first sperm fertilizes the egg, these cortical vesicles release their contents right into the space between the vitelline envelope, which is out here now, and the egg's plasma membrane. what the material from those cortical vesicles will do is it causes water to osmotically move into this space between the plasma membrane and the vitelline envelope, pushing it away from the egg. And so now you can see it. That was a terrible arrow, but that's the best I can do with the mouse. There we go. So it's pushing this thing out. It breaks the protein connection between the plasma membrane and the vitelline envelope. Osmotic influx of water pushes it out. And then finally, the secretions from the cortical vesicles chemically harden the vitelline envelope. And at this point, it's actually not called a vitelline envelope anymore. Once it's pushed out like this and chemically hardened, it's called a fertilization membrane. And that fertilization membrane, because it's been chemically altered, it will not let any additional sperm get in. They just can't, using their acrosomal enzymes, they just go boink, 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 boink. They can't get through, they can't dissolve through. So only one sperm will enter the egg. All right, this is the same as the previous slide. These are fertilized eggs, but these are fresh specimens, so they haven't been deformed at all. And what you can notice, I think, pretty easily is look at this fertilization membrane. It's a nice round sort of halo around the zygote. There's 1, 2, 3 fertilized eggs or zygotes here. And again, this is going to prevent entry of additional sperm, because keep in mind, out here, I'll circle a few. See all these little black dots? There's like 5 of them there. There might be 1 there, 1 there. Those are additional sperm. They're going to try and gain entry into the egg. Once that first sperm has fertilized the egg, this fertilization membrane forms. No more sperm can get in. Now, about the only thing I'd ask you on a slide like this, what is it? It's obviously fertilization. You can see the fertilization membrane. And the other thing I might ask you is, what's this structure right here? Well, that is a fertilization membrane. That's what you use to indicate that, hey, this thing's been fertilized. All right, this slide number 7. This is showing you what the embryo will look like shortly after fertilization. So these sea urchins we're working with, once fertilization occurs, you're going to see that fertilization membrane form, but then it's going to be about an hour. Again, the timing depends on the species you're looking at, but with these sea urchins, you're not going to see much of anything change after fertilization for an hour. Then the first thing you'll notice is, again, here's the fertilization membrane. It's nice and round in this one. This is a live specimen that was photographed. That's fertilization membrane. But if you look at the zygote, it's not a zygote anymore. This thing has divided. This first sort of developmental change you'll notice, it's referred to as cleavage. Now, cleavage is nothing more than mitotic divisions. And of course, mitosis occurs from early development until you're dead. Mitosis is occurring in your body now. What distinguishes sort of this early developmental division from other mitotic divisions is cleavage is defined or described as rapid mitotic divisions with no growth. You'll notice this fertilization membrane, it's rigid. It doesn't change size. So this thing is the same size as a zygote, this organism, but now instead of 1 cell, it has 1 here, 2 here, with a little cleavage furrow separating them right around here somewhere. These individual cells are called blastomeres. And so this is cleavage. Basically, if you remember the cell cycle, G1, S, G2, mitosis, it's like the cell cycle with almost no G1 phase. It's just divide, divide, divide. In the species we're working with, once these things start to divide, a cleavage division will occur about once every 20 minutes. So it's relatively fast. You go from the zygote to 2 cells, 4 cells, 8 cells, and so forth. So this is an example of cleavage, the developmental stage at this point. If I had that whole thing on a slide, what developmental stage does this indicate? You'd say cleavage. You can still see the fertilization membrane, and the individual cells are usually just referred to as a blastomeres. There's 1, 2 blastomeres in this specimen. This is a prepared slide. It's also showing cleavage a little later, like this one here has 1, 2, looks like 4 blastomeres, 2 in the front, 2 in the back. This one might have 8 cells or blastomeres. Now these are preserved specimens, so the fertilization membrane would still be present around this clump of cells. It would still be the same size. You just can't see it because it's too deformed on these prepared slides. Here is another example. This is a prepared slide, but again, right around here, that's a fertilization membrane. Now notice it's all deformed. That's because when you make these prepared slides, you're dropping the organism into a lot of different solutions, which can have some osmotic effects. You get, some stresses and strains, and so you deform some of these things a little bit. But that's still the fertilization membrane, what's left of it. And there's 8 cells in here, but they're the same size as the original zygote. So things haven't grown at all. This is just has 8 blastomeres in it. and it's still just a representation of cleavage. Again, this is still cleavage. It looks like 4-cell stage here. This is a preserved specimen, so they're a little prettier, but you can still see clearly on this one, that's the fertilization membrane. That's still the same size as it was around the zygote, but now you got 4 cells in it, one there, second one there, third one here. Looks like back here somewhere is a fourth one out of the plane there, but that's still cleavage fairly early, and that's really all you need to know. Now, after cleavage, I'm missing one developmental stage, but cleavage will continue. You'll have one zygote, which will divide into 2, and then you'll have 4. Sorry about the drawing. I don't draw well with the mouse. Then you'll have 8, something like this. And eventually you'll have this solid ball of cells. It should still be wrapped by the fertilization membrane. That thing is called a morula because it looks a bit like a mulberry, compound fruit, mulberry, blackberry, raspberry. They all kind of look the same. That thing will still be the same size as the original zygote, just has a lot more tiny cells in it. Now, after the end of cleavage, which ends at this stage called let me see if I can write it with a mouse, morula. Yeah, it looks like a 3-year-old did it, but that's the idea. That's the end of cleavage. What's going to happen now is the embryo is going to get bigger. It's going to grow. So this is the end of cleavage. Of course, mitosis is still occurring rapidly, but it's not called cleavage anymore. It's just mitosis. This structure right here is called a blastula. It's the next stage in development after cleavage. You'll notice there is no fertilization membrane around it. Basically, this embryo has expanded like a balloon. This inner spot here is filled with fluid, and that fluid, as it flows in, expands the whole embryo, and it basically hatches out of that fertilization membrane. I don't know if hatching is the right word, but that's the idea. So what you have here is something called a blastula. Boy, I'm not going to write anymore on these things. The blastula is kind of like if you've referred to at the end of cleavage, the morula is a solid ball of cells. This is a hollow ball of cells. There's a space right here on the inside that there's no cells in here. This is just hollow fluid with a layer of cells around the outside here. That's a blastula. The opening is usually referred to as a blastocele. So this stage is a blastula. If I pointed to it, what developmental stage is this? You'd say that's a blastula, obviously. And if I pointed to, what's this space in here? You'd say that's the blastocele, of course. And, that'd be about it. All right. This is kind of the last stage we'll probably get to see in the lab. The embryos start to die off because they're in a small amount of water and you have to keep them for 2 or 3 days to get much beyond this. But as I showed you in the previous slide, this is still a blastula, but this is a fresh one. This is one of the ones we'll look at in the lab. That's the layer of cells, and then there's just a hollow space in the middle here. So this whole area in here is the blastocele, and that's the stage that forms kind of a hollow ball of cells is how it's described, after cleavage. So pretty distinctive hollow ball of cells. All right, this developmental stage is called the gastrula. After you had, the blastula, sorry about all these names, I didn't invent them, which was a hollow ball, some of the cells will push in, they'll invaginate in and make this little tube that's almost like pushing your fist into a balloon here. You have this opening going out to the environment, and these cells grow in. That is what you're seeing right here, this funnel-shaped bit right here. That's where the cells on the surface here migrated in or folded in and make this little tube. This tube-like structure is called the archenteron. It's on your sheet of terms if you need the spelling, but the archenteron is the embryonic or primitive gut. This is going to be the digestive system eventually. One end's going to be the mouth. Eventually it'll fuse to the other side here. There'll be another hole. That's the anus. There you go, mouth to anus. That's kind of our design as well. So that is the archenteron. The opening to it here, or in my terrible diagram, the opening right here, that's called a blastopore. It's just extracellular fluid flows right in there. It's just outside of the organism now. Blastopore, archenteron. This space here is still called the blastocele. That doesn't change. So this is a gastrula. The process is called gastrulation. It's basically setting up the embryo's architecture. One end's going to be the head, where the mouth is. That's one opening. The other one's going to be the anus. So it's kind of setting up the main axis for the embryo. Just like with the previous slide, this is a gastrula. The process is gastrulation. You can see the cells folding in here. This is a live specimen micrograph of a live embryo, but the cell's folded in there, so this tube folding into the middle of the embryo, folding into or displacing the blastocele here, that's the archentron. It's going to be its guts, its digestive tract, and this is still the blastocele, and the whole thing is called a gastrula at this point. Gastrulation basically does a couple of things. One, it sets up the basic architecture for the embryo. You know, we don't look like a hollow ball. Excuse me. We don't look like a hollow ball. You know, we have a head end, a tail end, left and right side. That's kind of what you're setting up in gastrulation. And as we'll see in lecture, you're also going to set up the 3 embryonic germ layers in this process. All right, the next set of slides, these you'll be looking at in lab, but only as prepared slides. We are not looking at any live embryos of frogs, and we probably won't look at any live bird embryos either. So these will just all be prepared slides. It's not as many, but we'll take them a little later into development. We'll look at a few of the earlier stages in the frog, and in the chicken, it's mostly slightly later stages of development. All right, this is a frog embryo. Now, there are no slides of fertilization membranes in the frogs. The frog eggs are large, and basically where we start with the frog eggs is at cleavage. Now, this whole thing here, oops, let me circle it. This whole thing here, this is a slide, I believe it's described as early cleavage in a frog. And what you see here, these are sections, so they're not whole embryos, they're too big. So to see them on the microscope, they section them. So you got, looks like parts of 1, 2, 3, 4, and 5. You have 5 portions of 5 blastomeres in this slide. These are the junctions between the cells. This may be a bit of an artifact. That's a pretty large fluid-filled space there. Probably wouldn't be there. So these are blastomeres. All you can do with this slide, it represents early cleavage in a frog embryo. Now, the frog eggs are much bigger. The blastomeres here are much bigger as well. They have a little bit more yolk, which is going to affect their development, as we'll see in a bit. This is also a frog. This is cleavage a little later in development. You notice you have, there's a blastomere here, 2, a second one here, 3, and there's blastomeres, here, parts of them all over the place. Again, it's sectioned. This big crack here is an artifact. It wouldn't normally be visible on a fresh embryo. But a lot of cells in this frog embryo, late cleavage, these are still blastomeres. But what I want you to know, frogs have a little bit more yolk than the sea urchin or echinoderm embryos. And as we'll see, the yolk tends to interfere with the cleavage furrows, with division in the cell. The physical cytokinesis is slowed down by the yolk because it's fairly viscous and it's kind of inert. So what I want you to notice is, look at this cell here, for example. I'll kind of outline it roughly, and there's one here, one here. At this end of the embryo, then look at this cell here. Here's one cell here, and there's another part of a cell here, it looks like. You'll notice a difference, right? These cells are really large. There aren't many cells around this end of the egg. Few larger cells. This end, you have many more smaller cells. That's because of the polarity in the egg. The yolk, which is fairly dense, tends to sink down to the bottom of the egg. This is the bottom down here. The lighter cytoplasm will be up at this end. So because that yolk interferes with the cleavage divisions, you end up with an egg with polarity. This end of the egg, which has mostly yolk, is called the vegetal end or vegetal pole. This end up here that had very little yolk, more of the lighter cytoplasm, this is called the animal pole. The embryo, most of its development will be up here. These cells will be relatively slow to divide. They'll just end up being in the yolk sac for the most part. So animal pole, vegetal pole, this is cleavage still, and these are blastomeres. Another frog embryo, a little later, this is after cleavage ends, just like in the sea urchin embryos. After cleavage, you'll get the embryo will kind of expand a little bit in size, and it will be basically a hollow ball-like structure, and that's what you see here. This space here, that's fluid-filled, that's the blastocele, and the whole thing is a blastula, just like in the sea urchin. The only difference is, you'll notice there's a difference in polarity. All this mess down here, this is a lot of the relatively inert yolk, sits at this end. This is the animal pole up here, so there's a little bit of polarity with it, but that is the blastula of a frog embryo. Blastocele here, and the whole thing is just called a blastula. Now this is gastrulation in a frog. Remember in a sea urchin, I drew the gastrula, looks something like this. The cells just invaginate in like a little tube, and you have this, looks like a balloon that you punched your fist into. Sort of the same thing happens in this frog egg here, except all of this yolk, this is all yolk right here. I'll highlight it a little bit. All this is yolk. And the yolk is, again, fairly inert. It's kind of hard to move. It's like tar or something. It's viscous. It's not very active. It's hard to move. So you can't just have cells migrate in like a little tube here. So what happens with the frog, the cells just kind of start to roll in. They roll in at this point here, this direction. And if you're looking at the top of the frog egg, it wouldn't look like a hole. You'd see a little slit, kind of like that starts to form. That's looking down at this thing right here. The cells roll in and they form these little lips. This one here is called the dorsal lip. It's usually larger. And you see another lip here. That's the ventral lip. And the cells just kind of roll in. And you end up producing the archentron is actually kind of forming in this little space here. You can barely see it on this slide. Eventually, this slit will roll around until it does look like a hole. So it's kind of would look the same as this, except it's not hollow. The sea urchin, that's just a hollow opening. Here, you've got this big plug stuck in it. That is actually a yolk plug. They sometimes call this stage, rather than the gastrula or gastrulation, they call it the yolk plug stage. It's the same basic process. Cells are rolling in from the top into the inside, but they can't just move in freely because of the yolk, so they have to kind of squeeze in and roll over the edge here. So the cells are kind of migrating here, and then they roll in here, and they make this little slit, or they form this little dorsal lip. And all the cells kind of roll in, and it spreads around in a circle till you reach the opposite end. And then you have this, that's still basically the blastopore, but it's not really a pore. It's got a yolk plug sticking in it, so it's not a hollow space here. This is gastrulation in a frog embryo. All right, now this one's a little sideways. This is the dorsal aspect of the embryo. This is the ventral side. It's kind of been tipped on its side. I should have rotated the slide, I suppose, but I didn't. This is a later stage in development. We didn't look at this in the sea urchins because it really doesn't occur, but you'll notice this little indentation here. Now what you've got on this slide, it's a little hard to see, but there's a mass of dense tissue here. That's going to end up, or that is, it's a structure. It's one of the first structures to form if you're looking at an echinid, or if you're looking at a chordate, notochord. There. That's a piece of what they call mesoderm, one of the embryonic germ layers that condenses in the embryo fairly early in development. In fact, this is one of the first structures you can actually see. What's important about that notochord, it's going to be replaced in vertebrates with the vertebral column eventually. Even humans, we develop like a frog. We have a notochord early in our developmental history, but it gets replaced with vertebrae fairly early on. But that's a notochord. So the head would be kind of coming out of you, out of the screen, and the tail's going into your monitor or screen. So we've got a cross section of it here. That's a notochord. What they're showing you on this slide is something called neurulation, which we'll look at in lecture. But the cells up here, out here, it's what they call ectoderm. They form a thick plate right here. After the notochord forms, you get this plate of tissue. It's called a neural plate. And then it begins to sink in. You kind of see how you've got this sort of V-shaped fold right here. It's kind of a little V. That's actually called a neural groove, and these humps on either sides of the neural folds. The cells are sinking in here. Eventually what's going to happen, these cells will continue to sink in just above the notochord, and eventually they'll sink in far enough, and they'll close at the top. You'll have a little circle. You'll see it on the next slide. This is a process called neurulation. These cells back here are going to end up forming your brain and spinal cord. If you're a chordate, this does not happen in the echinoderms, just in the amphibians, mammals, reptiles, if you will. Right, this is the next slide in sort of the sequence. This is also neurulation, but those cells that folded in, that neural plate, you can see they've actually sealed off. They've folded in and just about closed at the top here. This circular structure right here, which is just above the notochord, which at this point is going to be about here somewhere. You can't really resolve it on this slide. But that structure right here will eventually mostly fill in with cells. At the anterior end, this is going to be your brain. and below that it'll be the spinal cord. So this is how the nervous system forms in only one phylum. This is a chordate phylum, which is what humans are in: frogs, birds, fish, reptiles, and so forth, amphibians. But this isn't going to be the neural tube. Now we'll look at its development in lecture a bit, but in lab you'll have to identify, this is the late neural tube. It's going to be the brain and spinal cord. This here, that's going to be the digestive system. So that's the archentron at this point. And the process is called neurulation. All right, the last, I think, 6 or 7 slides in this series. These are of chicken embryos. Again, these will all be preserved slides. We'll look at a couple of early development just because it's different. It'll give us some background for the lecture discussion. And then we will look at some much later stages of development, after the nervous system forms. We'll look at a few other internal organs in some of these embryos. All right, now the chicken embryo is a little bit different than the frogs or the sea urchins. Let me draw off to the side here. What happens in a chicken embryo, the embryo is actually just going to be on top of this thing. Then you've got this massive amount of yolk. Everyone's seen a chicken egg, I assume. Well, chicken eggs, reptile eggs, mammal eggs. Yes, they're mammal eggs. They all look about the same. There's a massive amount of yolk, which, as we've already mentioned with the frogs, it really slows down division. And then you've got this little bit of clear cytoplasm. It'll always be on the top of the egg. It's called the cytoplasmic island. That's where the embryo will develop. As we'll see in lecture, chickens, mammals, other reptiles have what they call incomplete cleavage. The yolk doesn't even divide. It just sits there. All the cleavage is going to occur in this little clump of cells at the top of the embryo. Now, this view you're looking at here, you imagine we're looking down at the top of a chicken egg. So the rest of the chicken egg would be off out here somewhere stretching around. So we're just looking at that little group of cells right here that's on top of the egg. Now, does it look like much? No. Our embryo starts off, if you could look at it in cross section, embryo starts off as a little, I should use a different color, a little flattened disc. And we're just looking straight down at that disc from the top. So this is equivalent to the blastula stage in sea urchins and frogs. Now they give it a different name in the vertebrates. It's called a blastocyst stage, or I should say in the mammals and birds, the blastocyst stage, not a blastula, but it's basically a solid mass of cells. We're looking, and you can't see much. It's just a mass of cells, kind of shaped as a little round, sort of flattened disc sitting on top of the yolk. This is equivalent. I actually should have said this is equivalent to the morula. This is just a solid ball of cells. There's not much to see. I think there's only one slide of this in our materials in lab. But that would be looking down at a chicken egg, basically at the end of cleavage. That's all you'd see. The embryo is just this flattened disc. Now, what you're seeing here, again, here's the embryonic sort of disc here. And there really isn't an equivalency that's good with the sea urchins and frogs, because our embryo isn't a hollow ball like it is in the frog and it is in the sea urchin. It's just this solid sort of flattened disc of cells. And again, we're looking straight down on the egg as if it were sitting in a bowl or something. The embryo, which is the animal pole, will be lighter. It always floats to the top. So this is that mass of cells. But what I want you to notice, right here, you'll see kind of a dark line of cells here. And another little group here. This structure, right here, that old dark line running down the embryo, that is called the primitive streak. This is gastrulation in a frog embryo. What's happening is the cells, they can't punch in because it's not a hollow ball. So all the cells on the surface are kind of moving this direction on this side, kind of migrating to the midline or sort of the equator here. So the cells are moving that direction. Over here, the cells are moving this direction. And what they're doing is they're kind of meeting at the middle. All the cells start migrating in, not all, but a bunch of cells start migrating in, and they stack up here and form these two big long lines. This is called the primitive streak. This is gastrulation in chicken and mammal and reptile embryos. What's going to happen is the cells are actually going to just push into the middle down here, kind of straight in from your field of view, just the way your vision is looking. They'll push in, And that's how they roll into the inside of the embryo. So this is a process of gastrulation in a chicken. It's called the primitive streak. It would be the same as a mammal. We'll get into some of the details in lecture. I can't draw on these things, so it would be kind of, it's easier to do with my lecture slides than with these actual micrographs, because while they're real, they're harder to describe. But this is the primitive streak or gastrulation in a chicken embryo. All you'd have to identify is the stage of development is gastrulation. You could call it primitive streak. And this structure here, these two dark lines, that is in fact the primitive streak. It's equivalent to gastrulation in a frog or a sea urchin. Now this is a later stage of development. This is kind of equivalent to the stage we're looking at in the frog, where you're getting the neural fold and neural tube forming. You see these dark, oops, let me go to a lighter shade here. These darkish lines going up here, kind of flaring out at this end. This one's a little deformed up here. These are the neural folds. Right down the middle, we're looking down at it, will be the neural groove. So this is a formation of the nervous system. The brain's going to be up in this end, and this will be the spinal cord. The other thing you can see is these little sort of segments of cells. I can't draw them very well. There's a lump there, lump there, lump there, lump there, lump there. Yeah, it's kind of, those are called somites. These are blocks of a type of tissue called mesoderm that'll end up forming the vertebrae and some of the muscles of the trunk, that sort of thing. But these are called somites. The brain's up here, but we'll see that better in another slide because this one's all collapsed and deformed. It would kind of look like a hammerhead shark normally. But this is just a diagram of neurulation, formation of the nervous system. The anterior end in this chicken is further along in development than the posterior end. Back here is still primitive streak. So the anterior end's a little bit ahead in development. All right, this one I like a little better. This is a little further along than the last one, but it's still a chicken embryo. And what you'll notice is, you see this part here, I don't know, it always reminds me like a hammerhead shark or something. This is the neural tube at the anterior end. This is the brain. It starts out as a bunch of little hollow sort of bags, brainstem, midbrain, forebrain, big hollow tube. This will end up being the brain of the embryo. It's kind of straight now. Later in development, it does roll up into the more compact form we're kind of used to seeing in a vertebrate brain, but that's the brain. Down here is the spinal cord. It's just better developed. This is I think this is a 33-hour-old chick embryo. You can see the somites here a little more clearly. Those little blocks of mesoderm. And I will mention this bit of the brain right here is going to eventually be the retina of the eye. It just hasn't pinched off yet. This bag-like structure you can see here, that's the heart. So nervous system forms pretty early in development. The heart is kind of a little after that. It's just this big hollow bag at the moment, hollow tube, but that's atria and ventricles of the heart. You can see the brain. Again, this is about 33-hour chicken embryo, much further along than the frog and the chondrum embryos we were looking at. This is a chicken embryo that's probably 72 hours old. It's quite well developed. You can see the head is now rolled up now. That's a cervical flexure, they call it. The embryo starts out as a straight thing, and then the head just kind of rolls up. So that's the brain. Right here is going to be the brain. A couple of things you can note. See this little round bit right here? That's the eye, and you can see a bit of the heart right here. But this is a well-developed embryo. It's going to have most of the organs at this point at about 3 days old. This is the same embryo, 72 hours old. It's just too big to get into field of view anymore. You can still see sort of the remnants of the somites here, these little blocks of tissue. Spinal cords running here. And what this slide is trying to show you is right here, coming out of the side of the embryo, those are blood vessels. They're a lot like our umbilical blood vessels that come out of a mammal's, umbilicus, your belly button. They call these the violin blood vessels, but these are going to take blood out away from the embryo to some gas exchange structures just under the eggshell. So these are actually they're comparable to our umbilical arteries and veins. They're pumping blood out of the embryo to pick up oxygen. Then it'll come back and supply the embryo with oxygen in a way to get rid of CO2 and so forth. So these are easy to see under the scope because they're so big, but that's I think you see a bit of the heart here. The eye was up that direction. Oops, eye was up this direction somewhere. That's a bit of the heart. But really this is just to show you the vital and blood vessels. All right, now this crazy thing, you will have a few slides that are referred to as serial sections because these later embryos get so big and they're so thick you can't see the internal structures. So what they do is they take an embryo, They embed it, usually in paraffin wax, and then they cut it into, thousands of thin, 5 micrometer thick or so sections, and they lay them out in order on a microscope slide. So right here, what you're seeing, this is one cross section of a chicken embryo at about 72 hours of age. You see part of a second one up here, part of the third right here. So they've sectioned the whole thing into a bunch of thin bits and just laid them all out flat. So these are in order, basically running from the head towards the tail. They just take a bunch of series of sections. Now let me just point out a couple of things you can see here while we're, while I have the slide up. This thing right here, that is the eye. You can see that's the lens right there, and that is the retina, which used to be part of your brain, but it ends up in your eyeball instead. So there's the retina and lens of an eye, lens and retina on this side. So this thing here, that's part of your brain. It will fill in with cells mostly. The only remnant of this hollow space will be the ventricles of your brain and the central canal of your spinal cord. So as this thing develops, this will mostly fill in with cells, so the brain won't be that thin. Your human brains develop exactly the same way. Here are the various chambers of the heart, like atria and ventricles. And now at this end, it looks a little weird. That's a spinal cord. And let me get my smaller pointer out. That little thing right there is much clearer than it is in a frog. That little circle right where that dot is, that little circle is the notochord, and above it, that's the spinal cord. Now, I can have the brain at one end, spinal cord at another. Part of the reason for that is the embryo at this point, let me scroll right here, kind of looks like this. If I can get a general idea, it's like kind of like that. So the brain's up here, spinal cord's running back here like this. When they do the section can be taken like this. So at one end, that's what we're looking at here. At the other end of the section, that's what you're looking at up here. So it's just the way the embryo is laid out is a little odd. In the lab, we'll have some textbooks that'll show you the serial sections, and they'll show you a whole embryo, kind of like this, and they'll show you where the section was taken. It helps you to orient, because these are kind of weird looking. They look like ink blots when you first start looking at them. Spinal cord, notochord, the heart's here. You got eyes, retina and the lens, you got the brain. That's about all I'd ask you on these. But again, you can see a lot more with your scope. You can scan up and down to look, look at the embryo from the tip of the head down towards the tail and see all the parts as you go. You don't have to remember a whole bunch of them, but I'll give you a sheet. It'll be posted on Blackboard eventually with the parts you have to know for the different developmental stages. But that's it for the slides for now. Take care.